I assume you guys can hear me. Let me make this full screen.
And yes, we can hear you.
Okay, good. So I'm going to tell you today about some calculations that we've done over various iterations of the sample contest organized by David Mobley and others, and in particular the sample 7 challenge. And I'm going to throw in a couple of other results along the way. So first of all, I always show a slide of my group in case I don't have time to get to it at the end. So most all of the calculations I'm going to talk about with regard to sample 7 were done by 2 people. Sherlock Shee here, who was an undergraduate, WashU undergraduate, did this as a summer project. She's now a first-year graduate student at Yale and will hopefully be doing a PhD in computational chemistry. And then Borna Novak up here, who was a rotation graduate student. He's an MD PhD student at WashU and did a summer rotation in my lab. Some of the other people that'll be mentioned along the way, Josh Rackers and Rose Silva, are both trained as physicists, and they're working on our next generation model after Amoeba. Ji Wang up here got his PhD with me at WashU and has stayed on as a postdoc and is our programming guru and has written done a fairly major rewrite of our Tinker software so that it's now blazingly fast on GPUs. Marie Lowry did some of the earlier sample calculations, and Chris Ho has helped us with parameterization, automation, and so forth. And then for the first time ever, I have to put in a disclaimer slide because this past summer, a bunch of us, the 5 founders shown there at the bottom, started a small company. We've all been friends for years. Ping Du Ren and I go back 20 years working on Amoeba. And we've set up a small company to take basically some of the kinds of results I'm going to show you today. And we finally decided that the best way to make this available and get it used is actually to try to do it through a company instead of just continuing to publish, academic papers here and there. So we've started up. These are the 5 founders. At the moment, we've hired a few people in France. We've really just started doing science. The plan is to open up a US office in Boston. At this point, in all honesty, that's going to depend on how COVID goes, but I'm hoping for maybe the start of the second half of 2021. So with that, we all know that most modeling problems in computational chemistry, drug design, what have you, kind of boil down to a trade-off between the model, the force field, the quantum mechanics, whatever you're using to model the system, and the ability to search the system, to search the conformational space or the flexibility of the protein involved in your system. And it's a trade-off between those 2 things, right? And so there have been a number of talks already at this meeting about various sort of search methods. And sometimes the models that are used, and in the context, again, of the current meeting, these are typically most commonly force field models. Sometimes it feels like, to me, those get shorter shrift. I mean, a lot of people, I think, kind of want to view force fields as something that are worked out by elves wearing green eye shades in a dark room somewhere, and people don't want to mess with them. But they're very important. And so in the what I'm going to show you today, the force fields are kind of going to be the star of the show. So we use, for the calculations, for energy calculations I'm going to show, we use a model that we've developed called Amoeba. And this is generally categorized by the field as kind of a polarizable force field. And indeed it is. We include polarization effects to account for the environment that a molecule or an atom finds itself in by computing induced dipole moments. But it's actually a little bit more than that. We think about half of the benefit of Amoeba is actually due to the fact that we use higher order atomic multipole moments on each atom. Instead of just partial charges, we have moments through the quadrupole moment on each atom. And that gives us a much better permanent description of the permanent electrostatics of the molecule. And that shows up in directionality of intermolecular interactions, things like directionality of hydrogen bonding, which is a very important thing. So polarization is important, that's giving you response to the environment, but the higher order electrostatics are equally important. So Amoeba is not just a polarizable force field, it's a little bit deeper than that. So over the years, we've parameterized water models, variety of organic molecules, ions, proteins, nucleic acids. We have further developments in the pipeline. Ping-Yu Ren and Jean-Philippe Pickmal are working on a so-called Amoeba Plus model that includes charge penetration. So once you include basic polarization and have an accurate description of the permanent electrostatics, the largest remaining error in force field models is due to charge penetration. And this is the idea that partial charges are not point charges on atoms. They're really representing electron clouds. And at close range, those things start to overlap and you need to get away from point approximations. And so the charge penetration is the way to do that. My lab has been developing a further model, which we call Hippo, which is this nasty acronym here, hydrogen-like intermolecular polarizable potential. In case you're wondering, this is a Hippo inside a hydrogen atom. And so the idea with Hippo is that it's actually even more explicitly fit to the physics. It's fit to sapped energy decompositions, and it uses the atomic multipole model, similar to what's in Amoeba, as an actual model of the electron density. And so we can include charge penetration. We separate the dispersion and repulsion effects, and because we use the multipole model as a model of the charge density, we can then derive a repulsion model based on that is now anisotropic. And we think that actually helps the validity of the calculations, accuracy of the calculations. And it contains a balance between polarization effects and charge transfer, the sum being what we might call the induction in the model. Okay, so that's a little bit about the force fields. So the free energy calculations I'm going to describe, I'm just going to skip over most of this. We use absolutely vanilla technology. So we're doing standard double decoupling with restraining potentials, and then we're doing lambda windows. And everything, importantly, everything I'm going to describe today is an absolute free energy calculation. It's not relative free energies, it's absolute, which is a whole different ballgame. It's much more difficult, we think. And so what we're going to do is we're going to decouple, disappear the ligand in its bound state until it's completely decoupled or annihilated. And then we're going to do the same thing in solvent, and we're basically going to take the difference between those two, and that's going to give us an absolute binding energy. And so on the plots I'm going to show you in a few minutes of our results compared to experiment, every point on those plots is an independent result, completely independent of everything else. It's an independent prediction all in and of itself. Okay, so our sampling protocol, just a boilerplate slide on our technology, we're going to use Amoeba. The multipoles are derived from high-level quantum calculations. Our bread and butter is to use MP2 with a correlation consistent triple zeta basis set. And we don't do that for fun. We actually think that that level is necessary to get the kind of electrostatics we want to get the level of accuracy that we're shooting for. Our goal is to compute absolute binding free energies within about a kilocalorie. We do annihilation or decoupling. We mix the two. We annihilate generally the electrostatics. The van der Waals we either annihilate or decouple. We also scale some of the key torsions in larger drug-like molecules. And generally it takes a total of 40 to 50 windows to do the bound and decoupling phases. And then we use a standard bar to compute the free energies between the adjacent windows and do error analysis by bootstrapping. The standard window length for most of what I'm going to describe today, there are a few differences. People could ask if they're interested. We use 10 nanosecond windows generally, and fairly standard technology. The calculations are run out of Tinker OpenMM. We have some new codes that we've also developed that are highly tuned for running Tinker and GPUs at this point. So with that, I'm going to describe a series of systems and just show you the kinds of results we can get. So the first one, this is an old system, but we never published it. So I'm going to show the results here. So this is from way back in sample 5, and this is a Q-cubitural based system with these naphthyl paddles at the end. So it's an open host system, and it binds molecules in this cleft. And this might look like a relatively stable binding pocket here, but really it's not, because those naphthalene groups at the end are moving up and down past each other like sushi knives. Okay, so they're moving up and down very quickly, and the binding pocket then is in the middle. And so these are experiments that were performed mostly by ITC and some NMR competition assays and so forth. And so there are a series of guest molecules that bind to this host, and they tend to be these acridine-like dyes and other similar dyes. And so the target host molecules are shown here. So you've got, you know, red, blue, green, orange. Must have been very colorful in Lyle Isaac's lab when they did this. And so we've got a total of, I guess it's 10 guests here that we want to compute. And so we use the protocol, the Amoeba force field and the protocol I described on the previous slides to compute these absolute binding free energies. And here's what we get. So this is calculated versus experiment. The red points are our predictions. 3 of the dyes, 3, 4, and I guess it's 5, have pKa issues in that their pKa is around the value at which the experiments were run. So you have to compute pKa corrections. But here are the results. The green bar is plus or minus 1 kcal between calculated and experiment. And so you can see we have an average unsigned error is inside a kilocalorie, and the you know, the correlation coefficient is 0.91, and the slope of a best fit regression line through that data, the calculated is equal to 1.08 times the experiment. So the slope is 1.08, and the offset's only about 1/4 of a kilocalorie. So, you know, that's a pretty decent result, we think. It's the kind of level that we're hoping to achieve. And in particular, we're very happy that that slope is close to 1, as it should be, and that the offset is relatively small. And so, so this is a post facto prediction. This was actually not entered in sample 5. And so, you know, you have to take that for what it's worth. So we've subsequently, when sample 7 came around, we entered sample 7, and there were 2 host-guest challenges that we entered here. One was for the so-called Gibb octa acids, which are these molecules shown here. I'll show a 3D picture in the moment. And so there are 2 slightly different variants depending on whether there's a carboxylic acid sticking out over here or whether the carboxylic acids are sticking vertically around the top edge of the system, as shown here. And then there are 8 different ligands. There are 4 carboxylic acids and 4 trimethylammonium ligands. You might guess that this one with the carboxylates more vertical and closer to the opening of the binding pocket, that might disfavor the carboxylic acids and favor the ammonium groups. And in this particular case, your intuition would be rewarded, as we'll see in a moment. And so this is a 3D structure here of the molecule. It sort of looks like an inverted lunar lander, for those of you that remember the Apollo missions back in the day. And around the top edge, there are these, what I will refer to as diphenyl ether linkages. There are these aromatic rings that are connected through a single oxygen atom, and that goes all the way around this top ring. And so getting the torsions right for that diphenyl ether linkage is what controls the flexibility of this molecule. It's breathing in solution, and so you have to get that right. And indeed, one of the messages of my talk, as everybody knows, is that you have to get the torsion parameters right. You can have the world's fanciest polarizable force field, and if your torsions aren't right, you will get the wrong answer. So the torsions are necessary but not sufficient. And so for that diphenyl ether linkage, if you do calculations on just the prototype diphenyl ether molecule, you get a 2D map around those 2 CO bonds that looks something like this. And it's difficult to fit that with standard 1D torsional potentials. So in fact, for our model, with the Amoeba model, we've used a torsion-torsion coupling term to include those torsion angles. And so here's an example. You can see that if the molecule, the host molecule is kind of squared off here at the top and relatively symmetrical, the binding cavity is larger, and that favors large guests. And if there's a breathing motion, largely resulting from rotation about those diphenyl ether linkages, then it can close up some, and this will favor smaller guests. And so the extent of that breathing motion is very important. Okay, so here are the results we get, and this is now a blind prediction. So this was entered in sample 7. This was our blind prediction result. And so again, you can see that, so there are two here. The one with the carboxylates vertical, that's the E series, and the numbers 1 through 8 are the number of the guest. The O series are the ones where the carboxylic acid is sticking further away from the opening to the host. And so the average unsigned error is 0.8 kcals. The R correlation coefficient is 0.91. Again, the calculated regression line is pretty good. The slope is effectively 1. The offset is again about 1/4 of a kilocalorie. So that's the kind of thing that we might hope for. You might note that the one molecule up here, the experimental result is 0. In fact, that particular carboxylic acid does not bind to the host with the vertical carboxylates on the host. And that's actually error on the part of the PI, Born and Novak, who did these calculations. He ran E1 here, and he came to me and said, Professor Ponder, it doesn't bind. And I said, well, the sample organizers wouldn't give us a molecule that doesn't bind. Go back and run it again. And he went back and redocked everything and ran it again and came back into my office and said, it doesn't bind. And so, I said, well, go run it one more time and really, stuff it down into the binding pocket and equilibrate it there, use some restraints to equilibrate it and then run it again. And he came back a third time and said, okay, that time it bound very weakly. And so that's what we turned in as the result. In fact, it doesn't bind. So, sometimes the PI has to get out of the way. So David Mobley, the sample organizers, ran the same calculations with a very similar double decoupling method using amber GAF, what they called reference calculations for the sample competition. And here's what they got. So that's the kind of result that, I don't know if that's completely typical, but that's what you get with a similar simulation protocol, but using a different force field. Basically, the big difference here between this slide and the preceding one is Amoeba versus amber GAF. Okay, the other system for sample 7 is more complicated. It's another Lyell-Isaac system. It's one of these Q-cubitriol-based systems with tryptosine moieties at the ends, and there are a series of 16 guests. And the Q-cubitriol-like systems tend to bind these organic ammonium ions. So these are all charged systems, organic ammonium ions. This is tricky because the host molecule has multiple conformations. So it can exist in an open conformation. It can exist in what we call an overlap conformation. Notice the pi-pi stacking here. And then it can exist in what we call an indent conformation. And the overlap conformation is in rapid equilibrium with a spiral situation where those two phenyl rings are moving up and down past each other. However, the indent and the overlap conformation on sort of the multiple nanosecond timescale we can access in simulations, they don't interconvert. So what we did is we computed the binding of all 16 ligands to both the indent form and the overlap form. The open form doesn't bind things. So we computed the binding of every ligand to this form and to this form, and then we had to weight those appropriately. So here's an example of guest 10. It's a diamantyl system bound to both the overlap form and the indent form, and here are the binding free energies that we came up with for those two. And you can see they're different. The larger ligands tend to bind better to the overlap conformation. The smaller, skinnier alkyl chain ligands tend to bind better to the indent conformation. And so what that means then is we computed the binding of all 16 guests to the indent conformation and all 16 guests to the overlap conformation using our standard double decoupling method. But now then, if we want to weight those relative to each other, we need to know the unligated free energy difference between those two conformations of the host when nothing is bound. And by the time we had to turn in the sample results, we were running out of time. And so we made a very crude guess based on just an empirical solvation free energy estimate of the solvation of the two conformations of the host. It was basically a wild guess. And we used that to get this horizontal leg to then submit our sample results. And here's what we submitted. Okay, so this is not up to what we like to think are our standards. In particular, we've got some bad outliers down here, and these are the larger guests. And so after the fact, we went back and did this, what I think is correctly. We used a series of an umbrella sampling calculation to rigorously interconvert the unligated, unrestrained indent conformation and the unligated, unrestrained overlap conformation. We ran about 50 windows driving distances and torsion angles to interconvert them. And what we decided in the end is that the overlap conformation is preferred by a little more than a kilocalorie. Well, that's not what was on our original prediction. The original prediction had the indent conformation favored by almost 3 kilocalories. So if we use this in umbrella sampling result to then weight the two series, the binding calculations to the indent and the overlap conformation, here's the plot that results. And so now then we have an unsigned error of about 1 log unit, 1.4 kcals. The regression line, the slope's not too bad. It's a little bit further off. The correlation coefficient's 0.83. So this is a harder system. It's very flexible. It's got multiple conformations that don't interconvert on short time scales and so on. So that's what we get. Again, there were reference calculations run with amber Gaff for this system. And here's what those look like. So, our results are maybe not as good as we might have hoped for, but they look like they're doing better than at least an amber Gaff sort of result. Okay, 2 other things I'll mention very quickly. This is a system that we, again, have never published, but it's one that we ran about 4 years ago now. This is another Q-cubitril-based system, so-called CV7, and it's a diamantyl system with a trimethyl ammonium group at each end, and the fit is perfect. It's like a hand in a glove here. And so all water in the interior is displaced, and this fits perfectly. Both the guest and the host are relatively rigid, so this should be a pretty simple system to sample. And this is unique because it is the tightest known host-guest system of this type. The binding energy is 20-some kilocalories. It's actually tighter than biotin streptavidin. And because both of these moieties are soluble in water, there are actually some people looking into using this similar to the way people use biotin streptavidin to fish things out of biological mixtures and so forth. So in the Isaac's lab, this is so tight that you can't do it in a single experiment. You have to chain together competition experiments using fluorescence, ITC, whatever, in order to get this binding energies this tight. They use 2 different chains of experiments, and these are their 2 estimates, 23.6 and 24.4 for the binding energy. And this is in quote unquote pure water with no salt added. If you simply add 50 millimolar sodium acetate, the binding falls off by 3.5 kilocalories. That's not unexpected. Something this tight is probably going to be very sensitive to salt concentration. So we ran this system to death. We ran over 5 microseconds of simulation in order to estimate this binding free energy, and we get 24.26 kilocalories. So in very close agreement with the ITC value, which is actually thought to be the better of these 2 experimental measurements. We didn't have to run it that long to get the free energy. The reason we ran it so long is we were trying to converge the enthalpy and the entropy, which are known from the ITC experiments. And those are much harder to converge, as everyone will know. We couldn't come up with tight numbers. However, it was very clear that the binding is largely enthalpically driven, and that's in agreement with experiment. Most of these Q-cubitril systems, due to the water displacement that goes on and how unhappy the water is in here, many of these are enthalpic for the major component of the binding. And then finally, I'll mention, so this is all fine. You might say, well, gee, you guys can do host-guest systems, but can you do a protein? And so I like to think that proteins are just big organic molecules, but you have to prove that. And so in the company now, and this is hot data, sort of hot off the press, we've just sort of started doing some science. We've run a series of calculations on bromodomain 4, and this is a crystal structure of a particular benzotriazepine ligand bound. So this is a very simple protein. It's a 4-helix bundle, very common class. It's a bit unusual because it's a left-handed 4-helix bundle. Most of them are right-handed, but there are some left-handed ones. And the binding pocket's always up at one end where the 4-helix bundle kind of splays open. So this is very typically where these proteins will bind things. And so some of you may know why we picked this system. So there are a series of 11 ligands that we computed, and Mike Gilson, who I think may be on the call, has done some calculations on these. This set of ligands was originally suggested by Philip Biggin, and it's very nice because for 10 of the 11, there are actual PDB crystal structures that the ligands bound. And it's a wide variety of ligands, you know, certainly not a homologous series. There's a wide variety of compounds in there. And so again, we want to see if we can compute absolute binding energies. And so we recently, Francis Jing in Pingyu Ren's lab, and he's now doing kind of an internship with Qubit, ran all of these, and here's what he gets for the absolute binding energies. And so again, we have an unsigned error that's 0.8 kcals. The signed error is close to 0. Here's the regression line. Again, the slope's close to 1. There's a little bit of an offset in this case. That's largely due to ligand 8. It's a bit odd that that ligand 8 is the only one that makes me anxious because, of course, ligand 8 is Xanax. And we think we actually know what's wrong with it when we've gone back and looked at the torsion. We think it's torsion parameters again. And so we've actually reparameterized the torsions between the phenyl ring and the benzodiazepine moiety. You have to get those torsions are very sensitive. We've reparameterized that, and we're in the process of rerunning this. We have some hopes that it's going to move closer. But anyway, that's what we get. And so Francis has also run this with Amber, using Amber GAF. So it's Amber 99SB with the Shaw ILDN torsion modification and GAF 1.8 and TIP3P water. And here's what he gets, the blue points. Now that's a bit controversial because I know other people have run this system and we don't get the same results they got, and we're still trying to figure that out. But all I can say is that's what we get. So I think I'll probably leave things there. I guess my take-home message would be that we think that if you want to do absolute binding free energies, we think the force field is important. There's a very large cancellation of error that's going on in a lot of relative binding free energy calculations. We're convinced of that. Everybody knows that, but some people may not realize how large that cancellation of error is and how much it's contributing to the results. And so, we're hopeful that if this technology for computing absolute binding free energies, if it's going to work reasonably well, that this can then be used to allow people to do things that look more like scaffold hopping and true drug design, where you can actually literally sit down with a blank piece of paper and design a molecule and compute its binding energy. That's the hope. And so, thank you, and I'll leave it there.
Thank you, Jay, for a great talk. Sorry, I'm frozen on my end, it looks like. There's a few questions that are in the Q&A at the bottom, so I'll go ahead and read probably 2 of these before we move on to our next speaker. First is by Brian Raddick. I believe I'm saying the name correctly. These sample results are quite impressive. You mentioned that the GPU implementation of Amoeba is quite fast. About how long does it take to get 10 nanoseconds of MD for a single alchemical window on one GPU? Is it hours or is it days?
Oh, it's hours. It's maybe 6 hours.
Okay. And then the next question is from Kiri Kidi. Why does polarization in the force field play such an important role in the results?
Well, because it's including the effects of the environment. So we think that, I didn't have time to go into it, we think that the reason we do pretty well on some of these host guest systems is maybe not even so much the description of the ligand, it's the bound water. So when you displace water, if you're off by 1 water molecule in the binding cavity of some of those Q-cubitrils or octaacids, your binding value is off by 3 or 4 kilocalories. And the water in the interior of those hosts is very unhappy. Okay, and it's unhappy in large part for reasons having to do with polarization. You're in a different environment. So, I think that's a potential real problem for non-polarizable models. They have trouble describing the water in very different environments. And unless you can do that, you're going to have, I think, big systematic errors in a lot of your force field calculations. The relatives may be better, but they're going to be big systematic errors.
All right. Well, thanks, Jay. You have, I think, about 6 more questions as of right now to answer. So please answer those in the Q&A. And we can go ahead and move to our next speaker.